So I think I'm online again, and I'm just doing a little bit of housekeeping. I'm able to hear myself. So, hi, hello everyone again. Welcome to another Saturday live stream. Microbehunter here, Oliver here. And today I'd like to do a little bit of microscopy together with you. And I see that several people have already joined in. And I'm just checking if everything is working as it should. The chat is working as well. I can hear myself, so I think the sound should also work. Okay. Yeah, and let's get started. Yeah, and today I've got a few topics again on my agenda that I would like to show you. Some of them a little bit more successful than others. So I'm also going to show you some of the things that did not work out during my experimentation. Sound is perfect. That's very good. Thank you. And during the first couple of minutes, I usually just say hello through all over the world until people start to join in, because it does take a couple of minutes for more and more people to join in. Okay, and so there is a hello from Lebanon, from New Jersey, from Canada, from Canada again, from Belgium, from Nicaragua, from Rhode Island, the United States, from Germany, USA, Illinois, Glasgow, the UK. Okay, sound is perfect. So here we are. Okay, let me also quickly check how many people have actually from India, from the UK. Okay, so let me quickly check how many people joined in up to this point, and there are right now, yeah, 46 people. That's quite nice. Yeah, community is slowly but steadily growing. So from the Netherlands, okay, California, India, very nice. Okay, so first of all, I have a few things on the agenda today, and if you've been a follower and a viewer of this live stream, then you probably already know that usually every live stream I dedicate to, let's say, one or two specific topics. Last week I changed the format around a little bit, so I'm going to show you several smaller topics, okay? And for those of you who already know the format a little bit, it's a little bit experimental. This means that I like to improvise. So sometimes things don't always quite work out the way that we hope them to work out, but I think we still can learn quite a lot in this whole process here. Okay, so yes, so what I would like to do today is the following. I've got a few cyanobacteria in here. So I would like to show them to you, what they are. I think they're quite a nice specimen. I would like to also show you a couple of old pictures. Actually, one of my first pictures that I made over 20 years ago using a webcam. And I would like to also show them how they basically compare to the original slides here. I have also wanted to show you some of this where I already made a video. So maybe you already watched the video. But look, I'm going to go a little bit into more detail. You don't see it now. This here is a little bit of moldy bread, okay? So we're gonna have a look at some of this stuff here as well. I did make a video of this, but I would like to spend a little bit more time on this today in the live stream. And I also, I would like to cut and microtome one of these leaves here, which is pretty difficult to do, and with only limited success. So I'm going to show you some of the difficulties that there are, and for this reason, I'm going to, I've ordered a new microtome, and I would like to test it with a new microtome as well, which did not arrive yet, but maybe in an upcoming live stream I will do that. So another important announcement, next week there will not be a live stream, okay? I just want to inform you on that. I'll post a couple of regular videos, but next week I'm not around, okay? And for this reason, you'd have to wait again to see my live stream in 2 weeks again, okay? So there's a question here, will these live streams be available later for reviewing again? Yes, the answer is clearly yes. In my YouTube channel, all of these live streams will stay. So if you go to my YouTube channel and if you check the tab with the live streams, you're going to see all of them from the previous year. I do not erase any of these live streams. They're all viewable online, and as a matter of fact, there are more people watching the live stream afterwards than actually during the live stream itself. Also has to do something a little bit with the time difference. So before we before I continue to talk, if you have any questions about anything microscopy related, or if you want to hear my opinion, please feel free to ask. because there are sometimes people also chatting a little bit in the chat. If you have a specific question to me, then please indicate this by putting an @ at Microbehunter or at Oliver. And we can also see our comments. Yes, the comments also will stay. I think usually it takes about a day or so for the comments to appear. The live stream is visible right away, but then YouTube takes a little bit of time to process everything, and then the comments will also appear, I think, a few hours or a day later. So, but nothing is lost. So so what I've got here is actually this sample over here is a very old water sample. I have to honestly tell you, I don't even remember where I got it from. Maybe already 6, 7, 8 months old. It's a pond water sample, and I kept it in this tube, and every time when the water evaporated, I filled it up with a little bit of non-chlorinated tap water. And and this is how it looks like. It looks pretty messy, pretty ugly, but actually I think it's quite interesting what we're able to see in here. And this is where I found some cyanobacteria, which I would like to show you. Okay, and I would like to talk a little bit about those cyanobacteria as well. So what I'm going to do is I'm going to simply take a pipette, and I'm going to go in here, and I'm going to go around a little bit and hope that I'm able to find something here. It looks very clear, the whole thing. Sometimes what's also useful is that you simply grab some of the gooey stuff here, right? And go a little bit like this, and then that's probably a little bit too much. So if it's too dense, it's also difficult to see, and it's not only cyanobacteria what we have here, but also other forms of other bacteria and algae and whatnot. Now I have a difficult time doing this with one hand. And I'm going to put the rest back here, and cover glass goes on top, and then let's have a look. Okay. And I was actually a little bit positively surprised, because there were quite a few of those actually in the sample. And as a matter of fact, I made a video as well, which I'm going to publish soon. So it goes on here. Usually I would like to remove any excess water, and the reason is because the cover glass should be quite flat. I do not want it to float, because I'll be going up quite a bit with the magnification, and therefore the working distance is quite small. So let's have a look, okay? And then I'm going to comment a little bit. No, that's the wrong microscope. That's the one, okay? You can also see a little bit on the top what I'm doing. Let's put it in here, and let's flip out this here. Let's flip out this here, and we see a lot of stuff here, right? Oh, these are not just some, I don't know, there's some other form of biofilm, but what we're really interested in are those long, thin, fibrous, things there, those filaments, right? And these are cyanobacteria, and I think they're pretty fascinating. So we have to go up a little bit with the magnification. Okay, I have to also refocus again a little bit. And let me quickly also pull out just a second. Yes, let's give it a little bit more light. Okay, you can always see the magnification up there, right? And let's go up, because at a higher magnification, we might be able to see something quite nice. Okay, look at those filaments here. And what do you notice? They move. Okay, so let's go up yet further. There is, of course, other movement as well. There are a whole bunch of ciliates in there, right? But what we have here, these are so-called cyanobacteria. They used to be called blue-green algae, but they're not algae. And I think that they belong, or could belong, to the genus Oscillatoria because of their movement. Okay, so cyanobacteria are photosynthetic bacteria. So these are actually, they're not algae. Because algae are eukaryotes, they have a nucleus, or at least a true algae. But they used to be called blue-green algae, and still are called like that, because in the back in the day, when they did the classification, they kind of dumped everything that looks like green, living in water, doing photosynthesis, and in filaments, they said, okay, these are algae. But in reality, they are not related to the algae at all, to the true algae that is, but they're actually bacteria. Yeah. So this is quite fascinating. There are a couple of comments here. By the way, again, an interesting, before I answer some of the questions here or comments, there are some people who actually said the following comment about the live stream. It says, yeah, the live stream is they like watching it, not so much for its scientific content, but because it's relatively relaxing. So that's also a little bit the how do you say, the motivation that I have is I want this to be a somewhat relaxing, Saturday live stream and not overload you with scientific facts, but a little bit of science is also fine. So there, the comment here is that they are deadly for humans, and the thing is that's depends a little bit. I wouldn't say deadly really, but there are actually some cyanobacteria which produce cyanotoxins, which are pretty bad. But not all of them do that, right? So, however, some cyanobacteria can be, they produce toxins that are not good, right? So I don't know about those here specifically, but I'm not swimming in this water, so I'm not so much worried about that. And I've noticed, okay, that's more related to succulent plants. I've noticed a few succulent plants' leaves have stomates on both sides, top and bottom. Is this common, you think? Is this specific to a succulent? Wow, that's an interesting question for which I do not exactly know the answer. I mean, I know that, for example, water lilies, obviously, they have the stomates on the top surface, but that's something that I can find out. Yeah? So that's... so I'm just reading again. So that is the question good morning. So the very long straight line in my sample, it moves, could be a cyanobacterium, and is it one cell? So let's have a look here. I'm going up a little bit because these filaments here are actually made of multiple cells, and I would like to zoom in a little bit more. And we try to find a couple of specimens here. You know what I'm going to do? I'm going to go and switch on DIC, just because the colors are so nice. Yeah, and I have to also switch on an arrow, and I'm going to now show you a little bit. If you look very carefully, then you can probably see that there are these subdivisions here, right? Yeah, so these are individual cells. So it's not the whole thing, is not one cell. It's actually made of hundreds, if not thousands of individual cells. But actually the subdivisions here, you see that these are the individual cells that are all connected in one row. Yeah. So this is actually like, this filament here is actually like a colony. Yeah. And what is kind of interesting here is that they still move in a coordinated way. Even though these are actually individual cells here, it still behaves a little bit like one single unit. So this is also a little bit of an interesting example where you see that the distinction between single-celled and multicellular organisms is starting to become a little bit blurred. Because actually bacteria are single-celled organisms. But again, one of these colonies, one of these filaments kind of behaves a little bit already like one separate individual. So the question is now where's the difference here? So now there is another interesting one. Are they moving because of the capillary action or by themselves? And actually, as a matter of fact, they move on their own. Okay, they will. What they do is apparently what they do is they secrete a polysaccharide, and then they have surface proteins that kind of push against the polysaccharide, and this causes their movement. So this is a movement that they're doing on their own, and because they're kind of moving back and forth like this, it could be the genus Oscillatoria. There are lots of species in this genus, but at least according to my identification book, it looks similar to Oscillatoria. A more detailed, now we're able to see it quite nicely, the individual cells. But a more detailed identification, of course, needs, you need to be a little bit more of an expert. I'm just a casual observer, and probably you'd have to do some biochemical or genetic tests. So what I would like, I'm going to go a little bit into the comments, and then I would like to show you a time lapse here as well. So yeah, I recently visited a favorite lake only to find it had taken over by cyanobacteria. Warning signs had to be put up to advise, from swimming. Exactly, you don't want to swim in a lake that contains that's green, filled with cyanobacteria because of those cyanotoxins, which actually might not be so healthy. Yeah? Thank you for the live stream. Is there any environment in particular where we could find volvox? Another question, is there any way of keeping cultures of hydra for a long time? Thanks. So volvox you can find, of course, also in ponds. I have to tell you that if you really want to have a highly concentrated amount of volvox, then you probably have to specifically enrich them by adding maybe some form of fertilizer to it. It's a little bit of luck which algae then will start to reproduce. So occasionally you're lucky and you're able to find them in a pond, but I would say it depends quite much on the conditions. Okay, so it's always a little bit of a surprise what you're going to find. So for example, I did not expect to find that many cyanobacteria in the water sample that I simply had standing here, right? Because they were not present in that density before. Yeah. Is there a way of keeping cultures of hydra for a long time? I want to quote one of my working colleagues at work who is keeping aquariums. I also used to have an aquarium long time ago, and he said that one of the big, apparently, discussions in the aquarium field is how do you actually reduce the hydra, because they are reproducing so quickly. So it's apparently difficult to get rid of them, right? So what I have done myself, I've actually also found a couple of hydra growing on some water plants from a lake, and I've been feeding them a little bit by adding some water fleas. And so they're pretty hungry, what I've heard. So you have to keep on feeding them with tiny water fleas or nematode worms and so on. And then basically, apparently it's possible to grow them. Huh? So it should not be too difficult, but I have not done this on an extensive way. Yeah? Another question by Caitlin. Do you use microscopes for a living with your job? I'm new to your channel. In my real non-YouTube life, I am a high school and middle school biology teacher. So what I do is in that sense, I use microscopes because, of course, I make videos also, and I show those videos also to my students. Because sometimes it's easier to quickly show them that what I want to show it to them than basically observing it directly under the microscope. I mean, of course, this hands-on thing in a school setting is of course something that's always nice, but sometimes, unfortunately, there's not always the time to do that. And sometimes, it's simply faster to pull out a microscope. We've got one with a camera connected, and I simply can show them the things that I want to show them, right? Of course, videos are also possible, and sometimes I do that, but you see, directly observing things always has a certain special touch to it, okay? Hello from India. What is the 1.25 written on the condenser? That's the numerical aperture, right? The 1.25 basically means that the objective, the maximum objective, the 100 times objective will also have 1.25, and the 1.25, let's put it this way, is kind of limiting. So if your objective has more than 1.25, which is highly unlikely because 1.25 is pretty much the maximum, then basically it's going to be limited by the condenser. So that simply has to do something about the numerical aperture. That's the standard 1.25. So what I would like to do now is I'm going to show you a video, a short one. Because those cyanobacteria here are moving, but look at this. I made a time lapse. So this is 5 times faster now. Okay, so I made this before, and I was kind of increasing the speed 5 times. And this is how it looks like. So you see the movement actually much better. Yeah, and you just gotta wait a second, and I'm going to. I've also added another clip here. Here, this one, for example. It's really when you speed up, the movement, and actually the becomes much more evident that the movement is actually not forced on the cyanobacteria by capillary motion of the water or water evaporating, but actually this is movement that they're doing on their own. Yeah, it's pretty almost a little bit creepy. Yeah, yeah, and this is apparently done somehow by polysaccharide being secreted, and there are apparently some surface proteins called pili, and they kind of push against the polysaccharide, and this apparently causes them to glide. Yeah. So what the magnification is under? I'm now using, or I've used my 60 times objective. So this basically means that with this magnification, pretty much any microscope that you have at home will give you a similar result. Yeah? My field of view of the camera, however, is very large. So this actually means that it actually looks smaller here than probably it would look when you look with under the microscope. Yeah? So, do cyanobacteria feed on anything else other than sunlight and photosynthesis? I have to tell you, they probably have to. Because the cellular components are not only carbon, hydrogen, and oxygen, but there's also nitrogen, there are phosphates necessary, and so on. And all of those other minerals need to be absorbed by the cell. Otherwise, they would not be able to make. Otherwise, they would not be able to make all of the cellular components. Essentially, they're photosynthetic, so the organic material actually is mostly made by photosynthesis. But all of the other minerals, of course, they need to absorb as well. Otherwise, they would not be able to make all of these other substances. Yeah. So I've recently gotten another new microscope, and my 4 times objective has a rainbowish ring around the lens when viewing down on my samples. How do I fix this problem? I don't know what this, honestly, sorry, here I have to say that I don't know. Okay, what do you mean also with a rainbowish ring around the lens? Huh. Yeah, so maybe there's some kind of an aberration that you're seeing, I don't know. Sorry about this, yeah? Okay, yeah, so I'm going to, yeah, power to you from Kansas City, okay, yeah. So science is cool. I hope that I'm not. Hope I'm not forgetting anything. Yeah. Okay. So is the movement of the bacteria random or coordinated? Let's put it this way. It must be coordinated, okay? Because it's evident that if it were random, then they would kind of be bouncing around, right? Like Brownian motion. Yeah, it is coordinated, and I think it's also fascinating that it is a coordination that it goes over several cells. But the question that I am having now, and I don't have the answer, is how do they actually communicate with each other, those cells, right? Or could the coordinated motion be a form of, I'm going to now say it, an emergent property. Yeah, so that maybe that this it appears to be coordinated because of the cells interacting with each other. Yeah? How many microns are cyanobacteria? They must have probably around a diameter of 1 micrometer, because that's approximate. I have to check with my micrometer slide, but bacteria have around a diameter of 1 micrometer. Okay, so I'm going to show you another one. This here are also cyanobacteria, which basically I also made this video here some time ago, which I also collected from a different pond, and also green, and they look vastly different, and they were actually floating on the surface of the water, and this kind of indicates this is not a pond that you would like to swim in. Right. I just wanted to show this to you as well, to show you that there is quite a huge biodiversity also in these cyanobacteria. Huh? Yeah. It goes away when the condenser is at the top. It's a rainbowish ring. The condenser should be on the top of a position because it's the normal position. So I suppose that this rainbow color could be maybe some kind of an aberration, or maybe some kind of a result of maybe some diffractions or so, color diffractions. So, but I have to see it actually to know a little bit more. Yeah, cells can communicate via osmosis or some similar sort of equilibrium mechanism. Okay, yeah, or by usually what they do is they secrete certain chemicals. Yeah, yeah. So let's go back to the cyanobacteria. Let's go a little bit down with. By the way, as another thing, I just want to mention here, if you look at these, this one, the live view now from the microscope, right? And if you compare it now to this one over here, you're going to see that the video actually has a much better contrast, and it appears to be much more clear and crisp. Yeah, and the reason is, and I do this actually with all of my recorded videos, is that I enhanced the contrast on the computer using my video editing program. Because sometimes people are asking, how do you get such nice crisp images, right? And the reason is yeah, the original footage often is not that very contrasty. It's like this. And then if you simply increase the contrast on the computer, it looks much better. Right. So what we have here is that is actually the way it looks, yeah, in original. And look, when I close the condenser, the contrast goes up, but also the brightness goes down. It's simply too dark, right? So that's yeah, also one of the little bit one of the explanations here. So yeah, you can a whole bunch of a biofilm. What I would like to do now is I would like to switch over, and I would like to go into phase contrast, because maybe we're also lucky to see something. So this is a DIC. I'm going to switch 40 times, and I have to also rotate the condenser. I have to put in the phase, and I have to open this here. I have to flip out the other one here, and here we go. This is now phase contrast. The phase contrast does not look quite as colorful, but it has another advantage. Maybe not here so much. But let's keep on looking. Let's keep on looking. Yes, yes. Now you have to watch really careful. Okay, that's what I wanted to show you here. I'm going to turn on the arrow again. Here we've got the arrow, and I would like to ask you to have a look at those tiny dots that you see over here. Some of them are a little bit elongated here in the background. Yeah, these are all the bacteria, right? These are all the bacteria here. They become visible quite nicely in phase contrast. And now, yeah, also these here, you see, this long one over here, yeah, all the bacteria. And now compare those bacteria to the cyanobacteria, which are green up here, right? They kind of look different, those tiny ones down here, right, compared to the cyanobacterium up here. Still, they're both bacteria. And they're both, in that sense, related, right? And back in the day, when people first started to classify those, what they found under the microscope, they simply said as well, the cyanobacteria, the green, yeah, they are basically algae, yeah, and because they're green and they do photosynthesis, yeah, and so they dumped them together with the eukaryotic algae, even though they are totally not related, yeah, just because. Two things are green and do photosynthesis does not mean that they're the same thing. Yeah, so and that's a little bit one of the confusions, so to say, or misclassifications that happened. Yeah, and this is the reason why those cyanobacteria originally still are called blue-green algae, even though actually they're strictly speaking not algae. Because algae, the true algae, are actually, they have larger cells, they have a nucleus, they're eukaryotes, right? So actually those cyanobacteria are related more to those, the other bacteria that you see here in the background. Yeah. So this is a little bit a thing. I just also want to share an anecdote with you. That one of my professors, this was a long time ago, when I studied microbiology at the university, and we talked about those, classification and so on, there was actually an anecdote that one of my professors told us, students at that time, and that is that it was a story about his own professor. So we're going back already quite a bit in time. And that in the middle 20th century, the cyanobacteria were reclassified. And they found out that those blue-green algae, the cyanobacteria, are not algae. This happened around middle of the 20th century. And the professor of my professor, when this turned out, was so unhappy about this. He was so totally shocked that essentially the cyanobacteria are not algae, that, and I'm quoting now, that he actually got ill. Because of that, yeah, and during his retirement speech at the university, he still maintained that cyanobacteria are actually algae, Yeah, it's a really an emotional thing. The people in the lecture hall were apparently already rolling their eyes. Oh, gee, yeah. But yeah, you can imagine that if somebody's spent his or her whole life researching those, and then all of a sudden they tell you, well, actually it's something different. Yeah, then this was actually a huge personal shock. Yeah. Maybe a little, I don't know, maybe a little bit of debate on naming as well. Should Pluto, the planet Pluto, be considered a planet or not? Yeah, some people also get emotional about these things. Yeah, just saying that. Yeah, nature is fascinating in any case. Yeah. So this is another one. I'm quickly going to pause again here, and I'm going to quickly go through some of the comments that we've got here, and then I would like to move on with the next topic. Okay. Let me quickly read. Is the movement of the bacteria, it's more or less coordinated? How many microns are signed? Approximately 1 micron across. Okay, this was again about the optical thing. What is the difference between this CH20I and your CH20? Well, actually my other microscope is a CH40 that I have, right? And this one is a different one. It's I've got this one is a BX53, so that's a different type of microscope. Yeah. So let me quickly read again. Cyanobacteria also looks thicker. Why is it that? Call of the cells. Why do so the question is why do the cyanobacteria look thicker and the other cells not? And I'm going to tell you the answer is, and I know this is not a satisfying answer, but I'm still going to say it. Why not? Why not? Right? It's simply biological diversity. Yeah? And there is actually a range of sizes, and some bacteria can be quite small, others can be quite large. Yeah. So it is simply a question of biodiversity. That we have, and if you look hard enough in nature, you're going to find all sorts of exceptions, unusual things, and so on. Yeah, so this is something that we simply have to get used to in biology. There is, as soon as you establish some kind of a rule, chances are pretty good that you're going to find some exceptions again. Yeah. Is cyanobacteria do not have complex cells? That is correct. Cyanobacteria being bacteria, they do not have cell organelles like the eukaryotes, like the other algae have. Yeah? Are they the only multicellular prokaryote? Now that is an interesting one. Okay, because the question is should you even count them as being multicellular or not? Okay, that's a question of definition here. You can say actually these are individual cells just sticking together, right? And however, cyanobacteria generally lack one important characteristic that multicellular organisms have. And this is that the cells here are essentially all the same. There is no cell differentiation. In a truly multicellular organism, like human beings, for example, we have a few hundred different cell types. Yeah. So there is no specialization. And I know that some of you are going to say, oh, what about the heterocysts? Yeah, there are indeed, and that's where I'm contradicting myself again, there are indeed some cyanobacteria where there are individual cells in between here which have, again, specialized function. So there is a basic form of specialization in some cyanobacteria also present, which is, again, a little bit a characteristic of a multicellular organism. So what we see here is that those cyanobacteria actually it's almost like they're in a little bit in a gray zone. They're kind of going already into the direction of being multicellular, but actually they are not quite as complex and as specialized to be considered truly multicellular. And the interesting thing here is that you see it's probably a question of us humans. We're always thinking in categories, right? Multicellular or not, right? But in nature there are gray zones, and yeah, there are sometimes no strict cutoff lines. Yeah. So and I'm getting carried away again here. Yeah. And you know what I'm going to do? I'm just going to move on slowly. Okay. So the level of organization is the cellular level of for cyanobacteria. Well, the level of organization here is that these are basically prokaryotic individual cells in a colony, and that there is essentially no formation of real tissues like we have in truly multicellular organisms. Yeah. Did chloroplasts first have a symbiosis with cyanobacteria? Well, actually, the first chloroplasts were cyanobacteria that actually moved into the host cell. We know that because of genetic analysis and genetic studies. Yeah. So this is called endosymbiosis. Yeah. So if you cover one side, will they all move to the light? I'd never tried that. That's an interesting one. The question is are they actually going to move towards the light? That is something I don't know. Okay. And something I might want to try out, but I think there might not be enough time to do this. Yeah. Yeah. So in any case, this is something that I kind of wanted to show you. I would like to show you another. What time is it? I always have to check my time. I'm already talking over half an hour, and I still have so many things on my agenda. Doesn't matter. I just want to show you. After all, it is. I'm not going to stress myself. We're going to see how far we get. I want to show you a second sample here, because in here I have got some real algae, real, true algae. So what I'm going to do is I'm going to take the slide away, and I'm going to just show this to you for comparison. It's the desk view again. Here we go. So I'm going to pull out some of those here. This is huge thingy. And I'm going to use my scissors. This is way too much. Let's cut it a little bit. Okay, so just that you see a little bit the comparison. Let's put the other one back so that you see a little bit the comparison between the cyanobacteria and some true filamentous algae, some eukaryotes. Okay, you're going to see that there are a couple of similarities, a couple of differences, but you might understand better why some scientists in the past decided to dump them together into the same group, algae. Okay, let's put them in here. So cyanobacteria here, the true algae over here, and let's have a look again. You want to have a look at the microscope. So these are the cyanobacteria. Where are they? Yeah, that's what we just looked at, right? Let's move over, and let's try to find just a specimen here as well. Yeah, and let's turn up the light a little bit here. Yeah, here are some of the. I mean, it's not the nicest sample, yeah, but you can also see that there are a whole bunch of filaments here. Maybe you have to go down a little bit with magnification. Yeah, but you see over here, there are filaments here. Let's try to find it. But it's a pretty old sample, so they don't quite look as nice. But let's this is actually a pretty ugly sample. But let's just stay here, okay? Yeah, and what you can see here is that the cells are of course much bigger here, right? And if you were to zoom in, then you could actually see that there are cell organelles and chloroplasts inside the cells. Yeah, so you see that there is the fact that it's kind of is growing in filaments. Yeah, and does photosynthesis, and it's in water, caused the scientists to dump them together into the same category. Nowadays, you do not classify organisms like this because you do not look at simply the color and the shape and how they live and their ecological significance, but you actually look at biochemical and genetic markers. Yeah. Yeah, so simply to illustrate this to you, the similarities and differences, and it shouldn't be surprising. Also here, not the best example that sometimes you can actually see some of those cyanobacteria actually growing on the surface. Yeah. So simply, I just wanted to show this to you briefly for comparison. Yeah. I'm going to quickly read through here. Yeah. Yeah. Yep. So again, this is not one of the nicest. I have to tell you, it's probably not one of the nicest specimens. It's also pretty old already, so there's quite a bit of decomposition also going on. Yeah. Yeah. Plenty of other stuff also present. Yeah. So, Okay, you know what? Let's leave the cyanobacteria behind, and let's move on to the next topic. Yes, the next topic would be, I just wanted to show you some old pictures that I made about over, I don't know, 25 years ago, 24 years ago. And I just want to give you a little slideshow, and I would like to explain a little bit of what I have done here. So let's quickly move this away. So this is kind of the next, let's put this away here. It's kind of the next thing, and I'm going to show you now some pictures, okay? So I will explain a little bit what it shows. And here we go. This is the cross section of the slideshow. After a couple of seconds, it's going to move. This is a cross section of a stem of a plant, okay? And I think it looks very pretty. And I'm a little bit proud of this because it was so much work to make. I can show you the original. I can show you the original slide as well, which I've got here. Yeah, this Cyclops and Daphne. These are commercially prepared slides. Okay, so why? And this is the cross section of a sunflower. Yeah, and first of all, why does it look bright on a black background? Very simple, because it's inverted. I simply inverted the colors in Photoshop. Yeah, so it's not dark field and no other fancy imaging technique. It's simply a regular bright field picture, and I changed the colors from positive to negative, and that's basically what I got, right? And why am I so proud of this? Why was this so much work? Because at that time, when was this? The year 2001 or 2002 or something like that. At that time, those modern digital microscope cameras did not exist yet. Or they did exist, but they were very, very expensive. I didn't have one at that time. I still took pictures. 2002, I made this one. Yeah. At that time, I still made pictures using film cameras. What I have, however, done at that time is I bought myself a webcam, 640 times 480 pixels. So it's a tiny resolution. And I put it into a plastic box and put it on top of a microscope into the photo tube without any intermediate optics or anything. So I had to refocus the microscope, and then it simply projected the image into the sensor of the webcam. And the thing is the following, that it was, of course, not possible to cover the whole specimen. So what I'm going to do now is, I think, maybe can I pause the slideshow here? I'm going to pause it now. And basically, what I had, the camera, I'm just going to trace out, maybe made pictures that were about this size. Yeah? And then what I did, and this took me several days of work, I assembled manually in Photoshop, I stitched together the complete image. So it didn't do this automatically, but I actually had to set the overlapping points, and then the computer put it together, right? So actually this was, I think, maybe several hundred images were combined together, right? And I just wanted to, this one also basically was made of several images that were combined together, and then I kind of, aligned it like this, cross-section of the sunflower again. And what I want to make clear here is that even nowadays, if you want to get a nice large overview of a picture, and if your camera zooms in too much, right, what you can do is you can take overlapping pictures and then you can stitch it together. And this is basically what I've done, right? So essentially, even though the camera had a very bad resolution, simply by stitching it together, I got a huge image, which basically looks very nice. Yeah. So I just wanted to show this to you. Why why I like this is not only because because it kind of looks nice, right? But because it was actually quite a bit of work, and I learned very much in the whole process. Yeah. I learned quite a bit. In Photoshop and stitching it together and making sure that the exposure is correct and so on. Yeah. Just wanted to share this with you a little bit to simply to tell you that it's sometimes not the camera equipment or the hardware that's necessary. Sometimes it's simply also the post editing that you're doing. So I'm going to again, as the slideshow is running, I'm going to go into the comments again. I hope that I'm not overlooking anything. Okay. Yeah. Yeah. Let me quickly. I noticed that fungi usually shows color in its reproduction phase and not in other phases. Why is that? Is it due to accumulation of nutrition for reproduction? Let's put it this way. Um, very often, not very often, it depends a lot on the fungi. It could be that certain fungi are producing certain pigments, and very often what you see sometimes, or I've seen this as well, that sometimes those parts that the spore-forming bodies sometimes are much darker than the rest of the cells, and it could be that especially if there are many cells clustered together, it simply becomes darker and it looks pigmented. It looks like it has a color because the individual cells of the fungus, the hyphae, might be simply too transparent. So that could be another hypothesis why some parts during reproduction look darker, because maybe there are simply many of the spores clustered together. Just a hypothesis, I don't know, but this could be maybe an explanation. Yeah, because I've also seen that sometimes those fruiting bodies, the spore-forming bodies, are indeed darker. Yeah. Yeah, so let me go here. How did you mount it? I guess you're kind of referring. I guess you're referring to this slide. This is a commercial slide. I'm just going to show you as well. Okay. So I did not make those slides. I actually just got ready-made commercial slides here. Yeah. So this is something I'm just going to show you, and you're going to see that the original. It's not exactly the same slide, but from the same series. So I'm going to go again into the scope view. It's kind of too bright. Let's move this out, and I'm going to show you now this how it looks like. And you know what? I'm going to go down with the magnification up a little bit here. This is, I need to go here. It's too bright. I need to focus. Yeah. And this is how it looks in original. Okay. Yeah. Here you see that because of the relatively large field of view, I could maybe stitch it together with, I don't know, 4 overlapping pictures, right? Like this 1, 2, 3, 4, maybe 5 pictures would be enough, right? To cover the whole image. But again, as I said, the camera was a small sensor, no intermediate optics, low resolution. So I think one picture was about this size, right? And I stitched them together in rows first, right? And then I combined the different rows. So there were a couple of hundred images. It was a lot of work. Yeah. So just wanted to show this to you, right? Yeah, how it looks actually quite different in original compared to what we've got at the end. Yeah. What's my favorite microbe? Oh, I don't have one. I usually, when people ask me that, I sometimes say Lacrimaria is one of my favorite ones because it has this long moving neck. Yeah. So, but there are essentially, I generally like things that are a little bit unique and interesting, yeah? So you did cellular mosaic photography like Mother Nature. Yeah, so I stitched a lot of images together, right? So just wanted to show this to you. I'm just checking here. And what time is it? I always have to do a little bit of time management here. Moldy bread and then plant leaves. Okay, I'm just going to show you a little bit of mold now as well. Because, I already made a video of this, but then I said, why not? Why not do a little bit more? Mold sometimes is also interesting. Huh? And I would like to show this to you here. And now you can actually see it also on top. I made this bread a couple of days ago, maybe 2 weeks ago. Forgot about it. And you see that there are all those gray spots on here. And those gray spots partially is mold and partially these are olives that are mixed into the bread. And then we forgot about it, or I forgot about it, and then this is how it looks like, right? And we now basically have it in a stereo microscope, and then when you kind of zoom in, then you're able to see a dense network of fungal hyphae. Hyphae are the cells, and those individual, and here what we see, the individual filaments that you see here are, of course, 1 cell thick. Yeah, so this is what there seems to be some dust and microplastics and whatever in here as well, right? Yeah, but essentially, if we want to have a better look at this, then we have to put it into the compound microscope. And I'm just going to do that as well, okay? Yeah, I'm going to read here as well. I bought my first micro photograph. It's a bit expensive, but highly recommended. Very impressive too. I previously bought a 5 megapixel camera and still can't get high focused photos. Why is that? Let's put it this way. It could be that essentially the image is actually in focus, but because the magnification is so high that it appears fuzzy. You always have to be aware that sometimes we've already reached the limit of what the microscope is able to resolve. And you simply expect more clarity, but actually that's the maximum what's physically possible. Yeah? So in this case, what I recommend is that you do a little bit of post editing, Photoshop editing, improving contrast and so on. Yeah. So, oh no, that's the bread. I'm just going to leave it here. I just want to actually pick the sample from underneath here because it also kind of looks a little fun. Okay, so let's do the following. I need some water, and I forgot a little dish. Okay, yeah, here I need some water, and I don't have a container. Just a second. Yep, here we go. Here is my water bottle. So I'm going to now do the following. I'm going to place a drop of water. Here, that's basically where my water is. I know the Petri dish is a little too large maybe. But let's put it away. And let's now do the scope. No, that's the wrong one. The stereo here. And with my tweezers, let's have a look here. So these are my tweezers, and look. Let's carefully remove. This takes a little bit of patience. Here we go. And let's try to put this now on the microscope slide. It's a little difficult to see. I don't know, is it on here? Yeah, yes, it's on here. You might not be able to see it, but I can see it. And let's put a cover glass on top. Yes, there is something on here. As always, I would like to remove any excess water. You can use tissue paper, or I'm just going to use my pipette again, because I do not want any instability of the cover glass floating around. So, and let's put it on the microscope. Okay, scope view is this one here. Let's put this away. Let's have a quick look at what we see here. And this is how it looks like. And again, we see filaments. Let's go a little bit higher in magnification. Okay, yet higher. Okay, and yet higher. Close the condenser a bit. Okay. And yet higher. 60 times. I'm not going to use my oil immersion. Okay. And I would like to show you, I don't know if I'm able to find this, honestly. But this was actually one of the examination questions when I got my university degree in biology. It was a commissional oral examination, yeah, about all sorts of topics, and One of the question was is how can you distinguish actually? Oh, these round structures, oval structures. These might be spores. But one of the questions is how do you actually know if you're looking under the microscope? How do you actually know if you're looking at bacteria, for example, like cyanobacteria, and fungi like you have over here? But the problem is this is not a good example either. I'm going to tell you the answer right away. Yeah, so is there any specific distinguishing characteristic that you should look out for? Well, of course, cyanobacteria are pigmented. They're green because they do photosynthesis. The fungi, they don't do that. Yeah. And one thing is, of course, that maybe we see it here a little bit, is that fungi are eukaryotes. So if you look actually into the cells, you might be able to see that there is something in there, right? These are cell organelles, the nucleus, sometimes there are multiple nuclei per cell. Yeah, but essentially there is something, yeah, inside the cell, but that was not the criterion here. Unfortunately, it's so dense that I cannot see it. I know you would like to know it, the answer. Yeah, these oval round structures seem to be spores as well. Okay, I'm going to tell you the answer. The hyphae, the cells of a fungus, they can be branched. Okay, and when bacteria, when they grow in a filament, they're generally not branched. So it has to do something with the way that the plane of division of the cells. Yeah? But unfortunately, it's so dense here that I'm not able to. I don't know if to see this, but yeah, fungal cells can be. I don't know. Maybe this could be an example here. I don't know. Yeah. Do you see actually here that there seems to be some kind of branching happening? Yeah, and that is something that we can see in fungi, but not not in bacterial filaments. Yeah. So I mean, I know. If you probably look hard enough in nature, you're always going to find exceptions. That's one of those. One of the things that they also told us is if you can think of it as possible, then probably if you look hard enough in nature, you're going to find it somewhere. Yeah. Yeah. But I just wanted to show this to you again here as another example of cells that appear to be in long filaments, right? So we looked at cyanobacteria, yeah, and here right now also the fungi. Yeah? So let me quickly go through the comments again. Fungal hyphae. Yep. I'm very interested in marine tardigrades, but there is very little footage of them. Is it because they're quite rare? Marine tardigrades. Do they actually live underwater? Yeah, I don't know much about them either, okay, unfortunately. But there's very little footage of them. It could be that they're, or simply not a lot of people actually made videos of them. Yeah, that could also be. Yeah, or it could be that basically people are studying them at university and research institutes, but not so many folks have actually made videos of them. Yeah. Fungi are eukaryotes. I didn't know. Yes, definitely, they are eukaryotes. They have a nucleus. Yeah. Does the branch is another cell? Well, that's now things are getting complex. So the question is it's an interesting question. It's a good question, but not an easy answer. The question is when it is branched, do you start another cell? It can be that a cell grows out and then it will separate later on, but there are different types of fungi. There are actually also so-called the aseptate fungi, where essentially the whole filament is actually one cell. Oh, look here. There seems to be branching up here. Yeah? No, or I don't know. maybe here, essentially because the cell walls in between them don't exist anymore. So there are different types of fungi. Some of them essentially have separate units of cells, and then there are other fungi where essentially it's like one continuous large cell with multiple nuclei, hundreds if not thousands of nuclei. Yeah, so these are the so-called the aseptate fungi. So you see how diverse nature is. Yeah. So I don't know about this one over here. It looks a little bit like it seems to be continuous, but then again, maybe I should go up with the magnification a little bit. Yeah. Thank you very much for doing this live stream. Thank you as well. Thank you for the thank you. Okay. Yeah, so this is maybe try going low magnification. Yeah, 60 times is indeed quite a bit. Yeah. And what we also see here is, that the thickness is quite different of the different cells. So it could be actually not one specific type, but maybe a mix of several. Yeah. By the way, again, in case you're kind of wondering why the background is so blue, this has to do something with the optics that I'm using. When you were looking at this, then it would look a little bit more like this. Okay. So that is this is a regular bright field. Yeah. So, and if you want to look at the whole thing in phase contrast, then it would look like this. Okay, and you see it's actually not quite as nice maybe. Yeah. So phase contrast is, I would say, has its values, especially if you want to look at individual bacterial cells. But because of lack of color and so on, it's probably not always suitable. Yeah, for every type of specimen. Yeah. Just wanted to show it to you. Marine tardigrades are difficult to collect because you need a large sample and a lot of centrifuge operation to find them. Yeah, this might be. And I can imagine, because tardigrades generally, I don't know about the marine ones, they like to cling to surfaces. So I can imagine that you have to probably collect some plant material or some other material, sediment maybe, where they actually cling to. And this could be also the place where they find food. Yeah. So. So there's a comment here. Multinucleate cells are observed. Yeah. So that's something I also wanted to show you a little bit. Yeah. Yeah. So and what time is it? It's already 1 hour. I would like to now again switch over, and I would like to show you something that did not work quite well. Okay. And I would like to do a little bit of, I would like to cut a leaf into a thin section. And because I was not so happy with the results up to this point, I'm still working on getting better results. And I even think about switching to a different type of microtome. Yeah. So, but I would like to show you a little bit of what I'm going to do next. Okay. So a little bit work in progress and a little bit, not so successful. But maybe you can be a little bit more successful. I want to show you, that basically I've got two of these microtomes here. I have to put this piston here back in here, right? That's one of them, and you can turn this here, and you can make very thin cuts. And the other one over here that I have here contains a clamp. Okay, so you can actually clamp the specimen here, and then you use a microtome knife, and you can make very thin cross sections. I would like to show you now what I prepared here. Now I want to use this one here today, and again with limited success only. And I've chosen a very thick leaf because it's easier to cut. And then I've made those carrot, how do you call this, pieces, using this cylinder here, which came with a microtome. So basically what you do is I simply kind of hole punched it out of a carrot, right? So I've made those things here, and they will be our holders, so to say. And they actually, you see, it's kind of diagonal here, so it should be a little bit straight. And what I'm going to do is I would like to make a cut into, just going to use a very sharp knife. It's my microtome knife. I'm going to make a cut into the carrot. And I'm going to put a piece of the leaf into this cut, into this, into here. And this will support the leaf. So I need to, I'm just going to use my scissors here. And I'm going to put this in, into the carrot. And maybe some of you have a better recommendation how to do this, okay? Because I was not happy with the result. Okay, but you get the idea. So it's kind of, and now this whole thing goes into the microtome. Okay, so let's put this away. Okay, and I'm going to put it directly in here. Okay, and it's all the way in. It's a little bit too deep. Let me focus this. Nope. And I'm going to turn it out a little bit. Okay, and it slowly starts to push it out. Okay, and now I've tried using a razor blade, but the problem with the razor blade is that they're too flexible. So, and I would like to show you a little bit the problem that I've had here and where I'm trying to find a solution. Okay, and of course you're going to glide across it. Normally I would fix this to my table. I cannot do that now because I've got a camera here. What I'm doing is I'm simply slicing off, and see, it's kind of very smooth here, right? And I will push it out carefully, and I will carefully remove this here, not cutting myself. And I would like to try it again. And sometimes it will actually work quite nicely. And I think right now I've gotten a small slice over here, okay, which I will put directly on a microscope slide with a little bit of water again, because I do not want this to dry out. Let me get some water. And the problem is that, of course, I would like to see it on the edge, on the corner. Okay, I'm going to make another one. And the problem that I have here is that, and maybe my technique is not quite right, but I tried so many things and it did not always work, is that when you cut across it, sometimes it will lift out the carrot a little bit, and the sections will be fairly thick. So this is a little bit of a problem. I need to make sure that the sections are more reproducible. Okay, and so sometimes the knife will also not catch anything and will simply slide over it. So let's try this again. Okay. So there are now 3 or 4 of these here. Separate this a little bit better. So, and I've chosen relatively thick leaves, and for this reason I hope that they're actually on the edge, right? So I will now try the following. I'm going to try to put a cover glass on top, as always. But I think that the cross sections are a little bit too thick. You see that there is even not enough water in here. So let's add a little bit of water. I think it's still way too thick. Okay, but let's have a look at it in any case. And probably it's going to be also too dark. And this is another problem that I'm having. And let me quickly go down with the magnification. So I need to get the scope view, and let's have a look here. It's actually worked much better than anticipated. Yeah, it's actually worked much better than anticipated. Yeah, but that's how it looks like. Okay. And I would like to do a little bit of biology, a short biology lesson again of plant anatomy. You see over here, it's kind of dark already, so the section becomes a little too thick here. Yeah. So this is one of them. You know what? I'm going to go down with the magnification. Let's have a look at another one. This one over here. Ah, yeah. It's a vascular bundle that I'm having. And I would like to show you then an example of how I would like to have it. Okay, here we go. So let's have a look here. At some of these things. So let's do a little bit of biology here. Question number one, what's the top surface of the leaf and what's the bottom surface of the leaf? Actually quite easily identifiable. Okay, here's my arrow again. Comment, I was told that when cutting with a razor blade, it's beneficial to clean the blade every now and then with a cloth soaked in ethanol. Yes. Sometimes adding a little bit of water also makes the blade slide a little bit better. Yeah. For the knife, I had the same problems. I was thinking about buying a low-profile disposable blades, and that's also one of the things that I'm trying to use. I've not tried it yet. There are disposable Microtome blades also available. Yeah. I have paraffin wax. I have not tried yet, but that is also on my agenda. The thing is when you're using paraffin wax, you need a solvent like xylene to remove the wax. And there are also safe alternatives. I try to avoid this because I do not want to work with, in my apartment, I do not want to work with xylene and other harmful solvents. But there are apparently also available alternatives that are not harmful. And that's something I've not tried yet. Yeah. So, okay, the pair. So maybe I'm going to, you've good results with the same microtome with paraffin. Okay, I will try that. Yeah. So, but let's have a look at the leaf here. This here, if you look here, this is the top surface of the leaf, and this is the bottom surface. And the question is, how do I know that? I know that because the cells on the top surface are vertically arranged. You see, it's called the cells are always, I would say, very often on the top surface vertically arranged like this. It's referred to as the palisade layer. And over here, you can see on the very outside here, there is a waxy cuticle. It's a thin layer of wax, which prevents evaporation of water. And then you have a thin layer of cells here. Maybe I'm going to go up here. It's called the upper epidermis. Oh, we can see the chloroplasts even. Nice. So here we go. Let's make it a little bit brighter. So on the outside there is a thin layer of wax which kind of insulates the leaf and prevents water loss by evaporation. Then you have a transparent layer that's called the upper epidermis, and you see that it's not green because it allows light to go through. Yeah? And then you have a layer of densely packed cells, and this is referred to as the palisade layer or the palisade mesophyll. And you see it's quite green because they're to catch a lot of sunlight, and you can see those tiny dots in here, many of them probably also chloroplasts. Yeah? So the top surface of the leaf optimized for photosynthesis. Okay, so we've got a waxy layer, we've got the upper epidermis, we've got the palisade layer, and then on the bottom side, let's move over here. So, what we have is we have a loosely packed layer of cells, and this is called the spongy layer or the spongy mesophyll. And there are air spaces in between here because carbon dioxide gas, which enters the leaf from the bottom, because there might be occasionally those stomates, the openings, right? And they will allow the air to go into the leaf. And then again here we have again a so-called a lower epidermis. Yeah? So you see that just by looking at the cross section of a leaf, you can actually see the different parts. Now, oh wow, this is also nice. And some of you already know what this is, obviously. What is this? This spiral-shaped thing here. That is something that was pulled out. That's xylem. Xylem are the cells that essentially transport water up a plant, and sometimes there are those strengthening rings in there. And when I was cutting it, I apparently pulled it out here. But that is very clearly, from xylem, which are the cells that transport water up to the plant and to the leaves. Also, for example, when you take a soft banana and put it under the microscope, you can also, they have also pretty long, spiral-shaped structures. Xylem, look how long this thing is. Yeah. So they basically prevent the cells from collapsing as water is transported up. Xylem. Okay. So yes, so we see also that there is some of these hair outgrowth. Yeah. I don't know if we're lucky enough that we actually are able to see also a stomate, an opening. I don't know about that. Yeah. So I'm going to answer a couple of questions here. Okay, so that's the bottom part here, and that's the top part of the leaf. And I'm going to show you now I'm also going to show you a commercially prepared slide as well. Yeah. Okay, bring back the biology days. Yes. I need a video that studies the biology of the blood, either interacts with the neurological systems of the body. Okay. Yeah. Okay, so you want to you need a video about blood. Okay. Is there a way to save the microbes after preparing them from a slide, even though there are billions of them are still removed? So what you can do is if you have a slide with microorganisms, okay, then I'll quickly show you the other slide, just a second. Where is the desk view? If you have a slide with any sample on it, also with microorganisms, and you would like to keep it a little bit longer, what you have to do is it will store for a long time, provided that it does not dry out. So you have to prevent it from drying it out. And what you can do is you can actually put it into a plastic container, which is moist. So you can actually make some kind of a support or a rack, so that it does not touch the water. And in the plastic container on the bottom, you have water, and it kind of rests on some kind of a holder, so that the water is not able to actually reach it. But when you put a lid on top, then the moisture inside the plastic container will prevent the slide from drying out. And if you do that, you can actually keep it for a fairly long time. Yeah. So yes, there is a possibility to actually keep them alive, so to say. Yeah. So I've done this already. I kept it already alive for several days already. Yeah. So what part of the xylem is the spiral thing? Well, the xylem is I don't know if I should actually make a drawing. I don't have a pen now, but the xylems are basically long tubes where water is transported upwards, right? And those tubes are basically empty cells. It's just a cell wall. Yeah. What you want now or what the plant now wants to kind of make sure that those cells, when there's water in there, it should not collapse together, right? So there's these strengthening rings inside there that kind of hold it apart. As a matter of fact, look, if you feel here, you've also got cartilage here, right? Kind of holding on the side, holding our airways open. Yeah, and that's basically a similar function the xylem has, is that there are these spiral-shaped structures, strengthening structures that kind of keep the cells open. Yeah, so that's we've got something similar as an analogy here. Yeah, so it's basically the xylem is basically the spiral-shaped structure is basically on the side of the cells. Yeah, kind of running on the outside. Yeah? So that's a little bit the thing. But let's go back here, the scope view. Yeah, we see a couple of plant here as well. Yeah, I want to show you now a commercial slide of a beech tree, and just to show you, I'm going to go here again, and I'm going to show you now. This is now a properly made slide. I have to find it first. Where is it? Here it is. Okay. And that's actually what I want to have. And that's, it's much thinner. Yeah. So, and it's also stained. Here we go. I'm going to open it more. Yeah. So we have a similar thing here. We've got the upper epidermis. You see over here the palisade layer, those vertical cells. Yeah. And you have down here the spongy layer, and now you can also see the air spaces. Yeah? So you see that this cross-section is significantly thinner. Therefore, we are also able to see the structures a little bit better. And then, of course, you also have the lower epidermis down here. Yeah? So you see that even though it's a different plant, yeah, those things here, this is where water is transported. The xylem and the phloem, for example, can be found in here. It's a vascular tissue, the vessels of the leaf. Yeah? But I just wanted to show this to you that it looks the same as the other one, with the difference that it looks actually much nicer. So it's significantly thinner, yeah, and therefore we're able to see the individual cells better. Yeah? Let's put this away. Yeah? Yep, I'm just reading some of the comments again. Yep. Is that red cabbage? No. It's it's called a beech tree, Beech, and essentially it was stained using some pigment. Unfortunately, it does not say which pigment was used, yeah. But I just wanted to show you that's kind of the thing that I'm kind of heading for is kind of a plant leaf cross sections that look a little bit like this. Yeah. So I have to somehow be able to make the cuts significantly thinner and more consistent. Yeah. So, and where is the other one here? Again, here we're again with the one that I made just before. Yeah. I need to, yeah, here we go again. Yeah. And here also because of the green chloroplast, so what I could do is I could try to destain the plant leaf cross section by adding alcohol, because the alcohol will actually dissolve the green chlorophyll, the green pigment. So, yep, get a leaf that has more stability, yeah. Maybe, yeah, this one actually is thicker. I've tried it with thinner leaves, but the problem is that the thin leaves are not quite as stable, and they basically sometimes fall over to the side. So I'm always able to see only the top surface, but I want to see actually the cross section. Yeah. Which stain can you recommend? Some months ago, I actually did make. There are several stains like hematoxylin stains, which are commonly used. Which is a red stain. Maybe that was actually used in the other one. I've also gotten some good results using methylene blue, because it stains the nuclei quite nicely. Yeah. So there are basic, depends on what you really want to stain. Yeah. If you want to stain the starch grains, you want to use iodine, of course. So hematoxylin, methylene blue, iodine works depending on what you want to stain. Yeah. 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 I think your slide is better than the stained one. Yeah, thank you. But it's a little bit. It's still a little bit thick. Yeah, and that's something that I would like to kind of optimize a little bit. Here, you see, it starts to. This is where kind of it got a little bit ripped apart. Yeah, and here it's too thick again. Yeah, but nicely visible here is the following. It's actually also quite revealing. Is that you see that the central part here is quite dark because of the pigment, the green pigment, the chlorophyll. But the upper epidermis here and the lower epidermis are white. So there are no chloroplasts in here, because the sunlight should go through the upper epidermis and hit the palisade layer, which catches the sunlight for photosynthesis. Yeah. So that is the thing here. I just wanted to show you another thing here. What I've also tried. This sample, what you see here, are also leaves. And they're not green. The reason is, and they're also over 20 years old in the container. A tulip flower. I cut the parts and I put it into alcohol, and this removed the chlorophyll, and the alcohol got all green. And then I exchanged the alcohol and I washed it several times, and now basically it's lost all of the pigments. Yeah. Just wanted to show this to you as well. So maybe in an upcoming video live stream, I can actually show you some more of those cross sections here as I'm staining them. Yeah, again, has advantages and disadvantages. You do not see the chloroplasts anymore, but you might be able to see other cell components better this way because they do not cover up the other parts. Yeah, because the chloroplasts are very green, so sometimes the specimen looks very dark. Yeah, but if you were destain it by putting it into alcohol, then yeah, other structures become better visible. Yeah. How did you find your passion or interest in microbiology? Okay, that goes back a long time actually. I know even as a kid I was always interested in the human body, and I always wanted to become a medical doctor. This was kind of a given. So I remember even in 4th grade elementary school when a teacher asked us, what do you want to become? It was very clear. I wanted to become a medical doctor because there was a book that my parents had, and this book was about first aid and medicine, and I really liked to read that a lot. And so I decided to become a doctor. And then when I graduated from school, I decided I did not want to become a doctor because I discovered the following: I was more interested in the biology part than in the curing of diseases part. This is kind of my interests kind of shifted a little bit. So this is why I decided to actually study microbiology and molecular biology at university. And yeah, I also had a toy microscope at that time, of course. So this kind of started off everything. But then later on, after I graduated with a master's degree in microbiology, I decided to go over into education. So I've been now a teacher for 25 years, teaching biology. And now I'll reveal something to you. I very quickly discovered that when you teach biology in a classroom setting with a chalkboard, and you have a syllabus to cover, and you've got a whole class of 25 to 30 students in front of you. Sometimes it's not so easy to do the real hands-on biology part, like microscopy. It takes time until 30 students have taken the microscopes and gotten ready. Half of the time is already over, right? So this is simply the practicalities of teaching in a classroom don't always permit to do interesting hands-on experiments. So this is when I decided some time ago, well, actually, yeah, I'm going to do a little bit of biology stuff online, live streams, YouTube videos. And I can also use that then also to show it to my students. And this way, with these live streams and YouTube videos, I kind of rediscovered a little bit the under quotation marks, the real biology part, which is the fascination and the love of nature and the observation and experimenting around a little bit. And not everything works, but then sometimes it does work. And sometimes there are some unexpected things that you see. And that's the fun part, right? And it's not advanced science what I'm doing here, but it's simply a kind of marveling at the beauty of nature somehow, and that's something that I kind of am missing a little bit, even though I'm a teacher. I'm missing a little bit in a classroom setting because I just have a syllabus to cover, right? And I have to assess the students, and there is not always the possibility to do the fun stuff, and sometimes because it also requires a little bit of skill, you want to do some hands-on activities with the students, and it doesn't work because obviously students are in the process of learning, and then you cannot make very thin cross sections because there's the danger that the students cut themselves, right? So you cannot use certain chemicals because they're toxic. So you see, you're kind of limited, and that's why I decided some time ago, okay, let's go online. Yeah. So a little bit of some background here. Yeah. So before yeah, it's already 1.5 hours again. So I'm going to quickly read through the comments again. Yeah. And then I think I'm going to call it quits for today. Yeah. Dry lens under the microscope. Okay, that's also an interesting one. Yeah. I'm blown away by those air spaces at the bottom side of the leaf. Never saw this like this before. Yeah, no, yeah, that those in the spongy mesophyll, those air spaces not so visible here because it's a little bit dense. But that is very important because this is where gas exchange happens. Yeah? It's also one of the reasons why leaves produce so much water vapor, because the water is lost over those air spaces, yeah, out the stomates into the atmosphere. That's also one of the reasons why, for example, in the tropical rainforest, why it's raining every day. It's because there's so much moisture being put into the air by the leaves of all of those plants. Yeah. What are those here for? Yeah, those here can have a variety of different functions as well. Sometimes they might protect maybe from insects or harm. And sometimes on the bottom side even, you can see sometimes not here so much, you can sometimes see a lot of here. And those here on the bottom side, they sometimes also reduce the loss of water moisture over the stomates, because they allow gas exchange from happening. The air can go through, but diffusion is limited a little bit, yeah, because of those. So sometimes over the stomates, over the openings, you sometimes in some plants you can see a lot of these tiny hair growing to kind of reduce the loss of water vapor. Yeah? Depends very much on the plant, yeah. But this is actually also, yeah, something, yeah. Yeah. So here. Yeah, interesting. Here can have many uses on plants, from deterring frost, breaking wind flow, and in some cases defense. Yep. Okay. Yep. And what I'm going to do now is, because I usually stop after 1.5 hours, I've covered pretty much everything I wanted to cover. I started off with cyanobacteria. I showed you some pictures that I made a long time ago. A little bit of moldy bread, plant leaf now, which the plant leaf actually worked better than I anticipated today in the afternoon. It was not that quite successful, yeah. And a little bit of Q&A. And what I'm going to, if you just joined later on, just an organizational comment. Next week there will not be a live stream, but again in 2 weeks. Yeah, I'll then think of again a couple of topics and a couple of specimens that I would like to show you. Yeah, this live stream today will be available online, and of course there are also regular videos that I'm publishing as well. If you're interested, I also put now a podcast that I started some time ago also into the YouTube channel. So if you click on the podcast tab, yeah, there are some audio only episodes as well about different topics. So I also put them on now to kind of put everything together. Yeah, and plenty of things, plenty of things. So I'm going to call it quits for today again. Hope that you kind of liked the episode, that you learned something. I certainly always learn a few new things when I'm doing those live streams. I wish you all the best. Hope to see you again, not next week, but again in 2 weeks, in 2 weeks' time. And yeah, happy microbe hunting. That's usually what I say at the very end. Happy microbe hunting as always, and see you next time again. Bye-bye.